So today's exploration will be a Ask Me Anything or Ask a Scientist. And we have three great speakers. So Emily Davidson as a student in environmental health and science. Milind Singh is a student in cell biology, and Mike Blazanin is a student in ecology and environmental biology. So I'm going to hand it over to Emily.
So as she said, I'm Emily, and I'm going to talk to you about how I became a scientist. So my talk is titled From the Farm to the Lab Bench, an abridged version, because it was a long journey. So I grew up in Applegate, Michigan, which is a super small town that's located right here in the thumb of Michigan. Population is about 265 people. It's pretty small. There's more cows than people, I'm sure. So here on the left is my parents' backyard. There's not a lot going on, obviously. And in rural Michigan, we spend a lot of time outside. So you can see here, my dad is living his best life with his puppy on his tractor. And my brother and mom are here outside in our backyard where we have a lot of bonfires and just kind of like hang out as a family. So in rural Michigan, there are not a lot of universities and not a lot of research labs. So you can imagine my high school experience was a little bit different. So I went to high school at Sandusky High School, which is home to the famed Thumb Dome, which is the largest gymnasium in the thumb of Michigan. It's located in Sandusky, Michigan, which is about 2,000 people. My graduating class had about 93. I loved band, drama, and English. I'd been singing since I was really young, and so I wanted to be an opera singer when I grew up. I hated math, physics, and chemistry, which is interesting considering what I do now. So my science education was actually not that great in high school. And to give you perspective, this is my high school biology book. It was published in 1995. I took biology in 2008. And the Human Genome Project, which was this large 13-year endeavor to basically map all of the genes in your genome by individually sequencing, was completed in 2003. So I really had no idea what was going on in terms of modern biology land. So I went to undergrad first to study arts and humanities. And when I left high school, I had been having some trouble singing and stuff because I had a lot of vocal issues. And I found out that I had a lot of food allergies. So I became interested in nutritional sciences, which is basically the study of how food is involved in physiological processes like digestion or in disease processes, essentially. And so I went and I decided I was going to study that and perhaps become a dietitian, which basically is a person who helps people figure out what they want to eat and what's good for them to eat, essentially. So I went to Michigan State and I realized I had to take chemistry, which kind of wasn't great for me because I didn't really like chemistry. And so I not only had to take something I didn't like, but then I'm stuck with, 500 students in the class. So I didn't do very well at the beginning. And so I had to go seek help from this person here, Dr. Amy Pollock, who basically helped me through chemistry 1 and chemistry 2 and helped me realize that I actually really enjoy chemistry. And so I stuck with this major and kind of got where I am today because I realized I really, really liked that subject. In addition to that, I was kind of saved by my academic advisor who suggested that I join an undergraduate research lab. And so I emailed some people and they said, okay, you can come volunteer in my lab. And that is this person here, Dr. Jennifer Fenton. So I worked in her lab for 4 years. I started volunteering by washing dishes, autoclaving, so cleaning things, organizing chemicals. I later acquired my own undergraduate research funding and worked on a project where we fed mice high-fat diets and studied the physiological processes that happen when you do that. And then in my last year, I worked on a project where I learned how to use these instruments here, which are mass spectrometers, which are a good way to measure all of these things called metabolites, which are these end products of metabolism. And basically, these end products of metabolism are all of these metabolic pathways. So I became super interested in how all of these pathways intersected to basically tell us something about physiology and biology. And so I knew that I wanted to kind of explore this interest more. So I actually took some gap years and I worked as a research associate at Colorado State University, where we actually assist researchers in the university and in industry to help measure these small metabolites in a variety of samples. So like samples from alpine lakes, and samples from mice, etc. And so I did that for 2 years, and I worked on this instrument here on the right, and that's me pipetting plasma. And then in addition to that, I spent a lot of time hiking in the mountains and a lot of time rock climbing, and it was an awesome time. So you can imagine when I went back to graduate school, it was a little bit of a shock. So that brings me here to Yale, where I study the pancreas. And so the pancreas is an organ that basically allows for the control of your blood sugar. And so here you can see if we eat a carbohydrate, we metabolize that carbohydrate like glucose. It enters the bloodstream. Your pancreas says, hey, I see this glucose, and it spits out a protein called insulin. And it basically allows for the body to take up blood or glucose and into tissues and lower blood glucose. So that's what I study. So in general, my research project tries to understand how antioxidants, which are molecules found in foods like tomatoes, etc., influence how the pancreas develops and functions. So you can see here we have a mouse pancreas. It's pretty funky looking. Then in the mouse pancreas, we have a little suborgan that kind of looks like this. And these cells here, which are the cells that actually spit out the insulin. And you can see that these cells look like this. And that blue, green stuff here is actually the insulin. And if we go a little bit deeper and we look at the same cells, in this here, we can see that the insulin granule itself actually looks like this. And that small little ball that's gray is the thing that gets secreted. So hopefully at the end of my PhD, we'll have a better understanding about how antioxidants influence how insulin is, spit out by these cells. So that's my story. And I hope you have some good questions for the little chat rooms. So thanks.
Thanks, Emily. So if you guys have questions for Emily, please put them in the chat. But I guess we'll move on to Milind.
Hey guys, you can see and hear me now. Let's see, move this thing aside. All right. So, see, my name is Milind. Today I'm going to tell you about my journey in science so far. There's a lot of time left, but let's see how it goes so far. So first of all, right off the bat, I want to tell you guys that I do science and I love it, but I do a lot of other things as well. And I don't spend all of my time in a room just doing science stuff. So an example, I love soccer and watching soccer. I think this is the Michigan University Stadium. I went a couple of years back, and I also love going to concerts and just having a lot of fun in general. But let me tell you where I'm from and my journey so far. So I grew up in India. So that's the country right here. It's a 15-hour direct flight. Go at your own risk. So I was born and brought up in the capital of India, New Delhi. And this is a picture of me and my family enjoying the festival of Holi, or the festival of colors. And during my growing up years, I used to spend a lot of time just video gaming and just gaming outside, like outdoor games and stuff. So that was a big part of my childhood. But a big part of my childhood also was developing a sense of a love for science and learning. And that was solely brought about by these amazing people here. So again, this is my dad, my grandmom, my mom, and my granddad. And all of them, from left to right, excluding me, were experts in these 4 different areas. So physics, biology, maths, and chemistry. And so like from a very early age, like these are some of the textbooks back home that, I studied in middle school. I just love to read and learn about science and maths. And these guys really helped me get a foothold into that. And as a consequence, I started giving Olympiads just for fun, just to learn and just have a good time. And in that process, I thought I would, just continue learning, sciences in the same way. And physics was one of my favorite subjects at this point. So for my next step, I decided to do my undergrad major in engineering physics or applied physics. And I joined this institute, the Indian Institute of Technology, for my undergrad. And it was a beautiful campus. This is a picture of my campus back in Guwahati. It was amazing. I spent an amazing few years there. However, during my second semester there, I had this amazing course in modern biotechnology, which was a common course then, and I was just captivated. This was the first time I had exposure to biology, and I was just my mind was blown. And I was like, you know what, I'm gonna take up a minor in biotechnology, study on my own, do some online coursework, and at the same time, I also decided to just email people and give a try to do research on my own, just like what Emily was talking about. So that's what I did. And just to highlight that point, I'm just going to tell you guys about the 3 significant experiences that I had during my undergrad. It cemented my love for research. That popped up later. But so in the summer 2015, which was at the end of my second year of undergrad, I worked with a professor in Bangalore, back home in India. And I studied how the nucleus in our cell, in the yeast cell actually divides properly during cell division. And this particular form of yeast is basically a brother of the yeast that helps in baking bread. And this is what I used to look at. These are the plates where the yeast used to grow. And it was a really amazing time. And the first time I had exposure to research. And that was the first time I had this gut feeling that, maybe this is something that I really want to do. And so in the next summer, I actually was again wanted to work in similar areas. I was very interested in cells. And so I worked with this professor at McGill, where I studied how microtubules, which are basically these rope-like structures that you can see in this one single microscopy image of a cell, how they help to regulate transport of stuff in cells. And again, I worked with an amazing team, had a more deep exposure of what it is to work in a lab, and loved every second of it. So I was like, hell yeah, let's work in my institute as well. So I decided to go to a professor at my college and work with them for the later half of my junior and my senior year of undergrad, where I worked on poultry viruses and studied their infection cycle. Here's me kind of working with the viruses in a secure way, with the hope to learn tips and tricks from them and apply them to human viruses. And we can clearly see how that is relevant in today's context. But this was again a great experience with a bunch of excellent people. And at the end of this, I was like, you know what, I'm just going to apply for PhD programs directly. I totally love research. Let's see what it is getting into grad school. And so that's what I did. And I'm going to use a Powerpuff Girl meme. I don't know if you guys have seen Powerpuff Girls, but whatever, to explain that. So while applying to grad school, this is what my application looked like. So I had a major in physics. I had a minor in biotechnology, had some biology lab experiences. But one thing that I was hesitant about at the beginning was interdisciplinary research, because I had worked in multiple areas. I was not sure how people looked at it, but it turns out, that's really well appreciated throughout the field. And certainly Yale does appreciate that. And that's how I got admitted into the Yale PhD program, the BBS program here, and pretty thrilled about that. And exactly actually 4 years ago was this picture, the first picture of me at Yale that was taken during the matriculation day ceremony. And so since then, I've now joined a lab here. I have joined Daniel Conor Ramos's lab, who is this professor right here. And we study 3 aspects of the brain. So the cell biology of the brain, how it develops and how it relates to behavior. And we use a really cool organism called C. elegans for our studies. And I can talk more about that in our breakout rooms. But I just want to highlight what I do. So I use these wonderful organisms to study how disruptions in energy production affects brain cells. And so it is known that our brain really loves food, and that's why you crave all that pizza and those burgers. And glucose in particular is a very brain-friendly food, so to speak. So I'm trying to study how the proteins that are involved in breaking down the glucose to provide energy to the brain, how they're affected in different conditions of stress. I'm putting these worms under stress and looking at how these enzymes behave in these different conditions, in different disease conditions as well, and trying to understand how the heck these things happen and what we can learn from them. And so for what's next, I don't know. It's not as simple as loading up a next episode on Netflix. It just doesn't automatically pop up. But I definitely want to contribute to science and create a community around me in a similar way that I'm working in right now. And so hopefully I'll be doing that and having a lot of fun in the process. And that's it. If you have any questions, let me know. But that's my presentation so far. Thank you.
Thank you, Milind. This is really interesting. We had two speakers that are kind of working on how glucose affects the body, but in different organisms and in different organs. Moving on, there'll be plenty of opportunities to ask them questions. So while we have time, Mike, if you want to get started.
Yeah, hi everybody. Hopefully you can see my completely black screen. So I do not study glucose, so just a little bit of diversity. So kind of going way back to when I was just a very young child. This is a picture of me when I was maybe 5 years old, and I grew up in the great state of Minnesota. And when I was even this young, but even especially as I kind of grew up into more middle school age, one of the things that I really, really loved to do most was to get lost in books. So often I would read fantasy and science fiction books. These are some of my favorite kind of book series, and I'm sure you recognize some of them, hopefully. And one of my really favorite things about kind of science fiction and fantasy books is that the authors create this world that is different from the one that we all live in, right? They create new rules, they add magic, they add new creatures, they create like this whole different reality. And then when you're reading it, you can kind of immerse yourself in that world and imagine, oh, what will the characters do when they have these powers or when these weird creatures exist in their world, right? And this kind of fascination and curiosity with, oh, what would happen if the rules were a little bit different has kind of remained a theme throughout a lot of my kind of academic journey. So as I got a little bit older and graduated into high school, a lot of my favorite subjects remained these things where there was a lot of kind of imagination and thinking about how do the rules work. So math and biology and economics were all things that I was really interested in middle school and in high school. And as I was starting college, these were actually three fields that I was all thinking about majoring in separately at one point or another, right? And so when I started college, I had no idea what I wanted to do, but I kind of liked these imaginative fields. And I eventually focused in on biology as kind of the slightly more favored one. And once I did that, I started exploring kind of different areas of biology that I found really interesting and that could be really fun for me. So for instance, I spent a summer in mice and kind of diseases that they get in their eyes. I spent a semester studying abroad in Venezuela, actually, in South America, where I studied like tropical plants and tropical ecology. And I spent a year working in a lab that was trying to kind of reduce pollution from runoff from farm fields using kind of different bacteria setups. But the area that actually ended up pulling me in the most was something that I originally had no interest in studying as kind of a real field, and that was evolutionary biology. And the reason that I never really thought that I wanted to do evolution was that my image of what evolution looked like was this, right? So I thought that evolutionary biologists, what they do is they go out and they find fossils, and then they study those fossils really, really closely to find very small differences. And then they put all those differences into these evolutionary trees, right? To kind of figure out what might have happened several 100 million years ago. And although there are evolutionary biologists who do that, this is not appealing to me. It just doesn't seem like fun. It's like, yeah, but it already happened like a long time ago. Who cares, right? And so when I was in college, what I ended up discovering is that there is a whole separate subfield of evolutionary biologists who do experimental evolution. And this is where you actually get to watch evolution happen in front of you in the lab or out in nature. So how does experimental evolution work? Well, usually it involves growing things in flasks or test tubes. And to walk you through an example of how this might work, imagine that you have some bacteria and you want to know, oh, how does this bacteria evolve under two different conditions? Say maybe a high temperature and a low temperature. Well, what you do is you put the bacteria into flasks, some at a high temperature and some at a low temperature, and you let them grow maybe for a day or two. And then you take some of those bacteria and you put them into a new flask, right? And you let them grow for a day or two. And you keep doing this over and over again until the bacteria have had a long time growing in these two different environments that we kind of created. And what I really love about this is it's just like the science fiction and fantasy books that I love to read, right? We as scientists get to decide what the rules are, and then we get to find out what the bacteria or other organisms are going to do under those rules. And what we get to find out is at the end, we get to compare how the bacteria have evolved to where they started. Right, so if you imagine that this is our original kind of ancestor bacteria that we started the experiment with, maybe the bacteria coming from the high temperature flasks evolved shorter tails, and the bacteria from the low temperature flasks evolves thinner bodies, or something like that, right? And you can really just see in 1 or 2 weeks right in the lab, oh, this is what happens when you put bacteria in a higher temperature, they evolve to be this way. Oh, well, what if they, what if we do some other different treatment, or we change the rules again, right? And this is what I do for grad school, and it is so much fun, right? So I take a bunch of bacteria, and now I even work with viruses. I combine them in different ways, and I put them in different treatments, and then I let them evolve in the lab for a couple of weeks, and I see what happens. And to me, this is just kind of the perfect way to live out my fantasy of all of those fantasy books of I get to just create fun experiments, see what happens, and then write up the results because it's meaningful for science. So if you have any questions about evolution or experimental evolution, I'd love to answer them, or anything else in general. But thanks, I'm looking forward to the breakouts.
Awesome. And there was another question about the picture that you showed us. It looked like the pancreas was covered in some sort of liquid. Could you tell us what that was?
Oh, yeah. So that's also a really good question. First of all, when you open the abdominal cavity of a mouse, there's all of this kind of like fluid that like lubricates the internal organs, which is just like, it's just some sort of like visceral fluid essentially. So everything is kind of coated in this like liquid essentially. And then also when I take the pancreas out, I do what's called a perfusion, where I take saline solution and I push it with a needle and a syringe through the pancreas to clean like all the ducts and all the capillaries of blood out of the pancreas. So when I go to look at it under a microscope after it's been put on a microscope slide, it's super clean. So there's no, red blood cells hanging around. It's not muddying anything up. So it's some of that residual liquid is from the saline solution. Yep.
Okay, interesting. Thank you so much, Emily.
Mm-hmm.
I'm going to turn over to Milind for the next question. Could you tell us a little more about what poultry viruses are?
Yeah, sure. So poultry viruses are basically viruses that affect birds or other forms of poultry in a very similar way as, viruses like COVID affect us humans. There are these other viruses which affect poultry, so hens and chickens and so on. But they don't do anything to us. So thankfully, or not much at least, so you can actually work with them. And but at the same time, they have a very similar infection cycle, how they infect the poultry cells, and how they grow and develop. So you can learn a lot from them, and relate them to human viruses. And so yeah, so that's what poultry viruses are.
Cool. Do you know of any diseases that are caused by poultry viruses?
You mean to poultry or to human beings?
Oh,
There's a mild yeah, so to poultry, I would say there are a couple, actually a lot of diseases, and it's devastating for poultry farmers. So it's something that they are very mindful of, and they have vaccinations and stuff, very similar to what we have to go through. So there are viruses that are similar. One virus is actually very similar to AIDS in humans, so that compromises the immune cells in the poultry. And cause them to die of other external infections, just like what AIDS does to us human beings, where it basically weakens our immune system, and then other viruses or other microorganisms take advantage of a weakened immune system and then affect us. To us humans, there is a link that one poultry virus that I studied in particular causes a very mild case of conjunctivitis. So that's something that affects our eyes, but it's pretty mild. So it's thankfully nothing severe for us humans. Glad, hopefully it stays that way.
Yeah, that's good to know. All right, one question we have for Mike. So you told us that you're studying evolution. Is there a specific aspect of evolution that you're interested in, that you're focusing on?
Yeah, so I got to chat a little bit about this in the breakout room, but so I'm really interested in a lot of evolution studies. I should back up. A lot of evolution studies are done with just one species, right? So you're just taking one species and you're watching how it evolves. But we know that bacteria, they don't live alone, right? They live in communities with many other species, right? Inside our intestines, in the soil, in the ocean, there are many, many hundreds or thousands of bacterial species all living together. And so I specifically study how does the presence of those other species change what evolution might happen. And so that's what a lot of my PhD dissertation research is on, that specific question.
That's super interesting. There's one more question I just saw in the chat. Do scientists normally leave bacteria in the lab for a few days to see if they evolve? Is that like the normal time span?
Yeah, I mean, it depends how quickly they grow, right? But bacteria grow usually about every hour they'll divide. That's like a pretty typical number. And so, if you want to study their evolution over sort of a short to medium term time period, you might do like a week, right? And in a week, you'll get about 40 generations of evolution, which is pretty good in terms of, the evolutionary scale. That's like the short end of things. There is one lab at Michigan State University that has been evolving bacteria continuously for about 35 years. And so they've actually hit 75,000 generations, right? Which is many, many generations. It's, I forget the exact comparison, but it's like more than the amount of time between us and chimpanzees when humans and chimpanzees evolutionarily split, right? So the experiments kind of span the range, but I would say usually on the order of like a month to a few months is very like typical.
Wow, that's really cool. Okay, so I think that's the end of the time that we have for the Q&A. So I'm just going to wrap up with a little bit of our outro, but thank you so much to all of our speakers. We really appreciated you guys taking the time to come and talk to us. And as always, you can check out previous videos from all of our other events at this link that hopefully someone can put in the chat. So you can watch any ones that you missed, or you can watch your favorites again. They're all there for you to check out. And that is all that we have for this week. Thank you so much, and we'll see you guys in September.